I weighed about 5 grams of corn flour and mixed it with 500 milliliters of drinking water. At this stage, the mixture looks cloudy and white. Next, heat it gently for about 5 minutes and do not let it boil. You can place the container in hot water, or if you have an oven at home, keep it around 60 degrees. Notice how the color changes from milky white to almost clear and transparent. This happens because heating starch in water causes gelatinization. The starch granules swell, burst, release amylose molecules into the solution, making it clearer, allowing iodine to react more strongly. Now take 2 test tubes and label them. Tube 1 will not receive saliva, this is our control, and tube 2 will contain starch, iodine and saliva. First, add 2 ml of starch solution to 20 ml of drinking water in each tube. This step cools and dilutes the starch solution. Because the amylose molecules reform their spiral structures in this solution, they can trap iodine very efficiently. This produces a strong blue-black color, which is much stronger than what we'll see with raw starch. Now set the wavelength to 580 nanometers. Adjust the height of the tubes just like we did in the previous lesson. Place the smartphone camera so that it records the blue color using color detector app. Now add 1 ml of iodine solution to both tubes and record the green channel intensity for tube 1 and tube 2. Now pause the video and think, should both tubes show similar intensity values at this stage? Yes, both tubes contain the same amount of starch and iodine, so the blue color should be the same. This initial reading acts as our blank. Now add saliva to tube 2. Immediately observe and record the blue color intensity. Continue recording readings every 30 seconds for the next 5 to 10 minutes. You already know why the blue color in tube 2 starts fading. Can you recall? Yes, it is because amylase in saliva breaks down starch, destroying the amylose spiral that holds iodine. When the solution is dark blue, very little light passes through. As the solution fades, more light passes through, and the app shows this change as increasing intensity values. This video is sped up 4 times. Between every 2 readings, I gently shake the tube. I'm careful not to change the setup during the experiment. Do not change the room lighting and make sure the screen brightness of your tablet or phone stays constant. After shaking, always place the tube back in exactly the same position. We started with blue channel intensity of 33 for tube 2. Immediately after adding saliva, the intensity increased to 45. By the end of 6 minutes, the intensity reached 178. Tube 1 showed no significant change. Pause the video again and think. Why did we set up tube 1? We'll come to this question later, but for now it's time for some maths. We'll use the Beer-Lambert idea from lesson 4, but with a small modification. Instead of comparing light passing through the sample to pure water, we'll compare light intensity at a given time, which is I, to the light intensity at the start, which is I naught. To make the graph increase as the color disappears, we calculate I divided by I naught. This keeps the values positive and makes the trend easier to understand. Here's a table of I divided by I naught values, which increase from 1.36 to 5.39. 5.39. 5.39 to 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 5.39. Using these, we calculate relative absorbance, which increases from 0.134 to 0.731. Now, draw a graph with time on the x-axis and relative absorbance on the y-axis. You can see that the absorbance increases gradually with time. The graph is steep only in the initial part, roughly from 0.5 to 3 minutes. This region is what we use to calculate the rate of reaction. Draw a best fit straight line through this region. Select two well-spaced points. Point 1 is at 1.5 minutes and 0.38 absorbance, and point 2 is at 2.5 minutes and 0.56 absorbance. Slope equals change in absorbance divided by change in time. So 0.56 minus 0.38 divided by 2.5 minus 1.5. This value represents the rate of amylase activity. If your graph shows a decreasing trend instead, that's perfectly fine. You can report the magnitude of the slope or describe it as a rate of decrease. You have seen how amylase slowly broke down starch in just few minutes. The blue color faded and we could actually track it using a smartphone. Isn't it amazing that enzymes are doing this kind of work every time we eat? If you want, you can imagine what would happen if temperature or pH changed. Just think about it. You can also design experiments to test these ideas maybe. Now let's connect everything we've done. Throughout this series, we used color as a guide, but what we were really measuring was light absorption. We compared how much light entered a solution and how much came out, and used that change to learn about concentration and reaction progress. That's the core idea behind colorimetry. Blanks, standards, absorbance, and the predictable link between absorption and concentration. These simple concepts form the foundation of every modern lab instrument. Light carries more stories than what our eyes can see. Stay curious, keep observing, and keep measuring.